Hello and greetings from Mumbai. My name is Dr. Arun Panda. At the very outset, I'd like to thank the organizing team of the International Multidisciplinary Conference on Current Research and Trends 2020 for giving me an opportunity to present my work on this virtual conference and on this forum. So I'm a craniomaxillofacial surgeon and I practice in Mumbai. My special interest is the area of facial plastic surgery and craniofacial anomalies. I also do facial cosmetic surgeries, facial feminization surgeries, and hair restoration surgeries. So my work, actually this work is actually related to hair restoration, but on a non-surgical modality. Okay, so let's go for the presentation. So the presentation title is basically the efficacy of adipocytes derived stem cells conditioned media in the management of Indian patients who are suffering from androgenetic alopecia. So basically this is an observational study which I have done, and then I have followed it up, and I am now following up with a study of larger subjects, the number of subjects. So which once the study is over, if anybody is interested, I can always share the results with them. But this presentation, this presentation is actually an observational study which we had done. Okay, so before I start how I have done the study, I would like to give you a brief introduction on what adipocyte-derived stem cells conditioned media is, and what exactly is androgenetic alopecia, and how we treat the androgenetic alopecia with ADAC conditioned media. So what exactly is stem cells? We all know that stem cells are basically the biological cells that can differentiate into any other cell types, and they can actually produce more of the same type of cells or different types of cells, or even the stem cells, as a matter of fact. Okay, so basically what happens is the stem cells, they have a huge potential to develop into any different kind of cell types in the body during the early period of life or during the growth phase. Now, so what is the basic difference between the stem cells and any other part of the body? So stem cells are basically, these are unspecialized cells. That means they have the capability to renew themselves by cell division whenever there's a condition which requires them to divide, even if it's after a long period of inactivity. So they can always divide and form newer cells, newer cell types, or they can form the new cell, the stem cells also. Okay, so basically there are two kinds of stem cells, embryonic stem cells and the non-embryonic or the adult stem cells. Basically the embryonic kind of stem cells are the ones which are actually done in the IVF, in vitro fertilization clinics and all, wherein the male sperm is actually, kind of fertilized with the egg, and then the zygote is formed and the zygote is allowed to divide, and then it is implanted into the uterus. So we are not basically interested in this. Now what we are actually talking about is the adult stem cells. So the adult stem cells, actually they exist throughout our body, and it right from the embryonic development, it is there in our body, every part of the body, and basically it is found in the bone marrow, the blood vessel, the skeletal muscles, the fat, and everywhere. Normally they stay in a quiescent state, non-dividing state. It can be for years together, and only when it is activated, whenever there is a disease or there's a deformity or where there is a requirement for them to divide, it starts dividing, and then it tries to heal that body. So basically they can divide or self-renew themselves, and it is indefinite. They can kind of keep on going on dividing as many number of times as possible till the person is alive. So what is the unique properties? As we just mentioned, that they are capable of dividing and renewing themselves a long period of time, and they're unspecialized, and they can actually give rise to specialized cell types whenever there is a requirement, whatever is this thing. So what are the source of the adult stem cells? They can be either hematopoietic or they can be mesenchymal. So the mesenchymal stem cells have actually come into a big vogue nowadays. So they can be derived from dental pulp, they can be derived from bone marrow, they can be derived from adipocytes, that's the fat tissues, or they can be from the umbilical cord tissues. Now, ADSC has actually come into a big limelight in regenerative medicines because of a few reasons. I'll just explain you. The first thing is it has got a huge potential capacity to self-renew itself. It can, kind of differentiate into different cell types like the adipocytes, the chondrocytes, osteoblasts, and other cell types also. The best part about it is easy access. We can always harvest the stem cells from the fat. The fat can be lipo-aspirated from the subcute layer, um, uh, and it can be easily used for the regenerative medicine. And it doesn't have the controversial thing which like the embryonic stem cells and all, because it's harvested from the same human being, and it can be processed and it can be used in the same human being. So there is no basically there is no controversies, ethical controversies related to it. Now, there are therapeutic applications. There are more than 130 clinical trials today which are going on and which are actually listed in the US National Library, and they have a capacity of paracrine secretion. Paracrine secretion means they have the capacity of kind of releasing these cytokines, chemokines, and the growth factors. Now, what exactly is this paracrine effect? It is very important for us to understand. Paracrine signaling means it is cell-to-cell communication. Unlike the endocrine factors, wherein the endocrinal signals are released from a cell and they have to travel for a longer distance. But the paracrine effect is basically the factors are secreted and it is immediately sent to the extracellular environment, and from there they travel to the nearby cells because of the gradient factor, because this cell is probably higher in gradient of that kind of cytokine, and this is of the next cell is of a lower gradient, lower concentration. So by gradient, it just flows to the next cell and it starts working there. Yeah. So what are the cytokines or the growth factors which are released from the ADAC? So we have the vascular endothelial growth factor, hepatocyte growth factor, the basic fibroblast growth factor, platelet-derived growth factor, keratinocyte growth factor, TGF, transforming growth factor, insulin-like growth factor, and other kinds of growth factors and cytokines. Now, coming to the point that how this ADAC is made. So basically, ADAC is made from the conditioned media is made from the ADAC. So the adipocyte derived stem cells, actually but with lipoaspiration, we take the adipocytes, the adipocyte tissues. The ADACs are actually isolated from the fat cells or the adipocyte tissues. And these ADACs are actually then, kind of cultured in a media. It's called the Adulbecose modified Eagles media, and it's left there for 72 hours in the culture media, and then it is taken. Now, what has happened here is from the ADAC, the growth factors of the cytokines have actually been, they have come out to the conditioned media, into the media, and that media is now flooded with the growth factors which we require, and that is centrifuged at 300 RPM for 5 minutes, and it is filtered, and those cytokines or these things are now taken and they are ready to be used. So that is known as the cultured, the conditioned media of the adipocyte derived stem cells. Now, the product which we are going to use has got this kind of concentration of the cytokines and the matrix proteins, the PDGF, the beta FGF, the keratinocyte growth factor, the transforming growth factor, beta 1, HGF, VGF, and other things with this kind of concentration, which I have just mentioned in this slide. Okay, now that's about the ADAC conditioned media. Now let's come to the hair fact. Since we're going to use this in the hair regrowth, let's just try and understand a little bit about the hair anatomy and how it works. So what is very important here, if you'd like to see, here is the dermal papilla and the matrix cells. Now the matrix cells are the cells which are sitting at the bottom of the follicle, and that is the cells which divide, and they form the hair, and the hair, and that keeps on growing up, and that forms the shaft once it crosses the skin level. Now, how does this cells divide? They divide because of the stimulus or the growth factors which are actually sent from the dermal papilla cells. So let's see a diagrammatic representation. So if you see in this image, so what you see here is insulin-like growth factor 1, IGF-1, and FGF-7 are the stimulating growth factors which are released from the dermal papilla here, and that is sent to the matrix cells in the hair bulb. Now, these matrix cells have got these receptors which kind of get attached to this IGF and FGF, and then they start multiplying, and as they multiply, they start growing up, and that is how a hair shaft is formed. It keeps on going up because it has no other way to go. So it has to grow upwards in the upward direction. That is how it keeps on growing. Now the hair goes into different cycles. We all know anagen, catagen, and telogen. Basically, the anagen is the growth factor, and the telogen is the resting phase. The anagen is the growing phase. Now, for a human hair in the scalp, there is about 1,000 days. The anagen phase is there, wherein the hair keeps on growing, and then after it's grown for 1,000 days, it goes into a catagen or the transitional phase and into the telogen phase. So once it goes into the catagen and telogen phase, that is where the growth stops. And then finally the hair falls off, and from the follicle the new hair shafts again. So that's the cycle which keeps on going for any hair over a period of time till the human or that the man stays. That's the diagrammatic representation of a hair growth. Now what exactly happens is, now if you're talking about the hair loss, which is mentioned as alopecia, there are basically the most important factor apart from the involutional alopecia or the natural way of thinning of the hair. The second most common thing is called as the androgenetic alopecia. So androgenetic alopecia, there is a reversal of the growth phase and the resting phase. So the anagen phase actually, which is for 1,000 days, that reduces, and the telogen phase or the resting phase, which is supposed to be very less, it starts becoming more, and the hair starts becoming thinner and thinner. So androgenetic alopecia is basically a genetic condition. It can happen in male and females both. So in the male, it is called as the male pattern baldness, and the female, it's called the female pattern baldness. So it's basically a combo of genes and hormones. Now the genes is basically, if the genes are there in a person, it is derived, it could be recessive or dominant. Only when you have the dominant gene coming into you, you start getting this androgenetic alopecia. And the hormone which is very important here is the testosterone. So testosterone is there in our body, and that gets, I'll just tell you how exactly, what exactly happens. So there is circulating testosterone in our body. Now what happens is, at the hair bulb, there is something called as a 5-alpha reductase. So this enzyme basically converts testosterone into 5-alpha dihydrotestosterone. Now what this 5-alpha dihydrotestosterone does at the hair bulb is it's got a very strong inhibitory action on the adenylate cyclase. So basically what happens is, because the cyclic AMP is the thing which gives the energy for any part of the body to work, and there is diminishing amount of cyclic AMP because the 5-alpha dihydrotestosterone, it has got an inhibitory action on this. So because of that, there is shortening of the hair cycle, and there is transition of the hair which are there in the anagen phase into the catagen phase, and that is why there is miniaturization of the hairs there, that is which is taking place there. Okay. That's about. So how do we classify? So we can classify the male pattern by Norwood's classification, or the Ludwig's pattern of classification is for the female baldness pattern. This is how we normally classify that. So what are the different treatment modalities available for androgenetic alopecia? So we can do hair transplant, wherein we redistribute the hair. So we can eliminate the triggering factors. We add multivitamins. We can give the finasteride or dutasteride, which blocks the conversion of DHT. And then the things which stimulate the hair follicles, like the minoxidil, the low-level laser therapy, the PRP therapy, macroneedling therapy, and the ADAC condition media, or the adipocyte derived condition media. So let's talk about the adipocyte derived condition media. There are today multiple amount of papers which are available, which shows the efficacy of ADAC condition media, which has mostly been done in Korea and all, but there are no studies which shows on Indian patients. So what we have done here is we have done this study on adipocyte derived stem cells condition media, the management of Indian patients who are suffering from androgenetic alopecia. So this is the product which we are going to use. It's from a company called Theraderm, which is manufactured in Korea. So it basically comes as powder and activator. So this is called the Biocell cytokines, which are injected into this thing, into the scalp, in the dermal layer. So the content is basically, as I said, the lyophilized powder, which contains basically the cytokines and the chemokines and the growth factors, the TGF3, vascular endothelial growth factor, KGF2, FGF7, PDGF, and beta fibroblastic growth factor. Apart from, there is a activator composition, which has to be mixed with the lyophilized powder to give the effect. So the BCC has got these important growth factors, the cytokines, which help us in the hair growth. The other thing which we are going to use is something called as a derma roller, wherein we do the microneedling of 1 millimeters. And what we are going to do is we are going to do an observation on the scalp by using a trichoscope, wherein we are going to count the number of follicles. So the subjects are basically, we have taken 5 subjects for our for our study, 3 men and 2 women of the age of 23 to 50 years, and we have classified them into the Norwood's classification and the Ludwig's classification. They were all systematically normal. There was no compromised patients. They were not on minoxidil or finasteride or LLLT. So we have not included any of these patients who were taking any of these things. And these patients were all treated and evaluated in my private practice in Mumbai. So we have taken a clinical examination we have done, medical history we have taken, and we have taken the informed consents, and we have told them they will be enrolled in the study, and informed consents have been taken. So these are the consent forms, the patient history, and we have taken the consent for the ADAC condition media. And this is the evaluation chart once we have done the patient. We are going to evaluate it for day 1 and day 60, day 180. So what's the treatment protocol? So we have what we're going to do is using this BCC hair serum or the ADAC conditioned media, adipocyte derived stem cell conditioned media. We're going to inject it intradermally every 15 days for a period of 3 months. So that is about 6 injections we are doing, and over a period of next 3 months we're going to observe. So total period is about 6 months. But the first 3 months we are injecting every 15 days. The next 3 months we are observing. So we're given a ring block, and then we have intradermally we have injected, and then we have done the injections. So basically, okay. So this is the media, and this is which is available here. Okay, so it actually, as I mentioned, it comes as the serum and the activator. I mean the powder, lyophilized powder, and the activator. So what we're going to do is we have to open. So this is how it comes. So we open the whole vial there, and what we do is basically this is how we open it. So this is the product. And then we take the syringe. I'll just forward this off a little bit. Okay, and then what we do is in the syringe, we fill up the activator. And this activator is basically, and then this activator is basically, now this activator is basically injected into the lyophilized powder, and it is gently mixed. And now this is all ready to be injected. So what we do is basically we take it. It's a very thick solution. So we take this, and this is what is ready to be injected. So we fill this in insulin syringes, and we put it in the insulin syringes, and it is ready to be injected. So what we do is basically we give a ring block. That's very important, because since it's a very thick product, it really hurts once we're injecting, and it burns. So basically we give a ring block, and that is how we inject. So once we are given the ring block, this is how intradermal injections are done. And then we use this 1 millimeter derma roller for derma rolling. And measurements are basically the 2 scales. We have taken the trichoscopic images to count the number of follicles before and after the treatment. And the second thing what we have done is we have taken the patients to know the patient how content they are with the treatment. So we have done something called as a patient satisfaction record. We have taken photographs, and we give the patient the photographs before and the day 1 and the day 180, and we kind of take the consideration, ask them to scale it up, how happy they are. So basically they would be scaling their satisfaction from 1 to 5. So these are the 2 things. So there are 2 patients that we have done clinical trichoscopic. So there is basically on the day 1 the count was 48, and the day 180 was 53. So there is basically an increase by 10.41%. There was no complication. Patient containment scale was 4. Second patient, this is a clinical photographs, trichoscopic images, results 40 day 1, 47 day 4, day 180, and there were no complications. And the patient containment scale was 5. The patient was very happy. Third patient, again day 1 and day 180, there was not much of increase in this, about 11.1% from 36 to 40. The patient was not very happy with this. Next patient, day 1, day 4, trichoscopic scans. Results was basically from 54 to 59, increased by 9.2%. The patient was very happy with this. So once we have done this, so we have taken this in the tabular form. The analysis of the trichoscopic images shows that there is an increase, an average increase of 11.38%, and the patient containment scale is an average score of 3.6%. Talking about the results, so we have taken 5 patients in the study. All the patients received the same treatment for PCC hair serum for 6 sessions every 15 days for 3 months, and then we have kept them in observation for the next 3 months. We have done microneedling therapy with the derma roller along with the treatment, along with the ADSC injections. And all the patients completed 6 sessions in 3 months, and then we kept them in observation for another 3 months, totaling of 6 months. And then we have done a trichoscopic images, and we have found that in an area of 3.14 square millimeter, that is a radius of 1 millimeter. So there is an increase in density from 47 to 52.6%. That's about 11.38%. And the patient containment scale was around average of 3.6%. In conclusion, I would like to conclude that though this is an initial observational study and it doesn't have the statistical number of subjects, but it has demonstrated really a great effectiveness in the hair regrowth for the Indian patients suffering from androgenetic alopecia. And best part is no adverse effects. Undeniably, we still require many randomized controlled trials for this kind of study, for this product. But I'm sure it has got a huge potential and it can be the first line of treatment for patients suffering from initial stages of androgenetic alopecia and the patients who want to go for the non-surgical modalities. Thank you very much. And if anybody has got any queries, they can always email me at arunpanda@reliefmail.com. Thank you so much.